Our next speaker for this session is Dr. Schaeffer. He's a professor in biology. Dr. Schaeffer is an expert in molecular population genetics. His laboratory focuses on understanding the genomic processes that alter gene and chromosomal frequencies in natural population of plants and animals. His lab is currently using next generation technologies which will help him to improve favorite fly Drosophila pseudobucera's genome assembly and annotation of gene models. Please join me in welcoming Dr. Stephen Schaeffer.
I'd like to thank the organizers for inviting me to speak today. By its very nature, I'm an evolutionary biologist, and as such, I'm a person that's interested in history, so I sort of frame my talk as a history of the study of chromosomal evolution, dating back to the days of Thomas Hunt Morgan, and then incorporating my own history into this process. Because DNA is a historical molecule. It captures the history in terms of the mutations that happen down lineages, and we can reconstruct that by looking at its sequence. And so my history has been tied up with the history of DNA. Now you may ask yourself, why in the wide world of sports would anybody work on a fruit fly? Melissa Rolls told us a little bit about working with fruit flies this morning in her talk on neuroscience and neurobiology. But when I go to the annual Drosophila meetings, people will see my tag in the fruit fly meetings, and they'll say, fruit flies, why do you guys work on fruit flies? I get them in my kitchen all the time. How do you kill them? That's the usual response. Well, it turns out fruit flies have been an excellent model system for the study of genetics since 1910, when Thomas Hunt Morgan first isolated a white-eyed fly mutant. But the main things that make this a great model system is that it has, you can control the crosses. We can't do that in humans. You can't say, I would like you and you to mate. I'm going to take your child when after it's produced, and I'm going to do things with it. That just doesn't go over very well. So Scott Selleck was telling us all about human genetic disease, and clearly that's a very important thing that we all are worried about, but you can't always work with humans in the way that you can manipulate systems to understand the underlying molecular biology. And so we can do controlled crosses. We have mutants in humans and fruit flies, but we can essentially do a better job working with a fruit fly model system. And then the big thing is fruit flies have very short generation time, and we can do a lot of things in a very short 2-week period from generation to generation. But I want to take you through the history prior to me, and then develop my history intertwined with DNA. So we've gone back in time to the lab of Thomas Hunt Morgan at Columbia University. It's not as fancy as what you would see here at Penn State. We don't have twines of bananas hanging in our laboratory anymore. We do have bottles of fruit flies in their media, but it was kind of it's not really high tech. But so much interesting biology took place in this lab. And this was due to this individual, Thomas Hunt Morgan, who won the Nobel Prize for his discovery that chromosomes were associated with the genetic material. And this white-eyed mutant was very informative because it was a sex-linked trait, and you could actually see that the biology traces with a particular chromosome. So that's what he won the Nobel Prize for. He had a stable of awesome students. If I could go back in time, these are the 3 people, including Morgan, plus Morgan, that I would want to meet. This is Calvin Bridges, H.J. Muller, and Alfred Sturtevant. These people did more with the genetic tools of their age, and we essentially, as biologists, are mopping up for them and basically demonstrating that with fewer tools, they were able to establish an incredible lineage of an understanding of biology. This is the first genomic map. It was basically something that Calvin Bridges built in his basement. It's essentially this four-sided tower, which each side represented a different chromosome in the Drosophila melanogaster genome. And as they discovered mutants, they discovered that they could put the genes on a map using a method called genetic mapping, where they calculated the distance between genes based on the process of crossing over during meiosis. And they were able to order genes in the genome. The amazing thing to me that Sturtevant and his colleagues did was basically, they not only looked at the genetic map of one species of Drosophila, they looked at multiple species of Drosophila and found that the maps were similar, but not quite the same. And the idea was that in the comparison, that you could find out that the same genes were on the same chromosomes, but they were in different orders. And we now know that how those changes occur are via a mechanism known as chromosomal inversions, where basically two breaks are introduced in the DNA and that the order of those genes are basically swapped. And so when you compare different species, essentially what you're looking at are reorganizations of the the genes that are on those chromosomes. In fact, we've had genome rearrangements in the human lineage. Two of our chromosomes from our common ancestor with chimpanzees have fused together to form one chromosome as opposed to a single one. And this is the main question that I'm really interested in. This is my academic grandfather, the person that you often hear cited as the person that said, nothing in biology makes sense except in the light of evolution. This is Theodosius Dobzhansky in his office, I think it was at Columbia University. History is important because it's not only that we have the history of particular experiments that have happened and the history of us, but there's also the history of ideas. And many of the ideas that we are researching today really emanated from these people in terms of the evolution of chromosomes. This is a picture of the marvelous polytene chromosomes from fruit flies. You isolate these from their salivary glands. And the thing that's amazing, we don't have anything like this in humans. This is another advantage of working with a fruit fly, because you get these amazing banded, puffed chromosomes, and you can actually see changes in the orders of the bands if you look carefully enough. Of course, the students, when they do this in the lab for the first time, are like, their head blows up. But essentially, if you look at the chromosomes long enough and you're careful with your analysis, you'll find that there are differences in the order of the bands. And by inference, we assume that those are different gene orders. Well, in this species that I work with, Drosophila pseudoobscura, Dobzhansky found, in collaboration with Alfred Sturtevant, a rich polymorphism for gene order on this chromosome, which is chromosome 3 in Drosophila pseudoobscura. And they named the different chromosomes after the places where they were originally discovered. So it's over 30 different kinds. So the question is, what is this all about? Why are chromosomes varying? Well, not only that, but if you go out into populations in the southwestern U.S. and you collect fruit flies from those populations, you can see differences in the frequencies of these different chromosomes. And amazingly enough, the places where you see major shifts in the frequency are also where you see ecological shifts in terms of the climates in the southwestern U.S. That pattern can be generated by a number of different evolutionary scenarios. One would be one where there's little migration, and let's say this red symbolized chromosome arises in that area and it doesn't really move around. So it initially is found here and it doesn't spread. So that would be the little migration model. Alternatively, you could say that this is driven by natural selection caused by adaptation to particular new environments. And that would occur if we basically look at extensive migration among populations. You can think of migration as a mechanism very much like diffusion. The more exchange there is, the more things will be, the frequencies of genes will be the same. It would be like having a glass of water with a dye in it, and if you have if you have an open glass that has just clear water and a glass with dye, and there's no communication between those glasses, they'll stay different. But if there's exchange of fluid between the two, they would eventually have the same color fluid. Now, that's a very brief history prior to me. There's a lot of other things that have happened. But on the left here is my history. And on the right is the history of DNA. And I'm starting here 3 years before I was born with the publication of the structure of the DNA helix by Watson and Crick. Now, I was born in 1956, and in the interim between the discovery of DNA and when I was, and a little bit after I was born, DNA replication was discovered. In 1964, I figured out that I wanted to be a scientist. That would seem like I was only like 7 or so. There I am in 3rd grade with Miss Stepperud, and I was a baby boomer, so we had really large classes. This is over 33 or so kids in my class. But this was an amazing year for me because a lot of things happened. Miss Stepperud was a bohemian-style teacher. She was into art, music, and she got a bunch of kids that were interested in science. And she didn't know what to do. So what did she do? She wheeled the TV in and let us watch rockets going off. So the space program that was initiated by John Kennedy was basically the backdrop for little kids like me, wondering if we could be an astronaut too, that sort of thing. The other influential person is this person. This is Jacques Cousteau. And during the 60s, they started these National Geographic TV shows, and they showed, Jacques Cousteau would be there, Philippe and I, in the submarine and that whole thing. And I got into science for that. The other thing I owe to Miss Stepperud is my love of classical music. She played that all the time, and I still listen to it today. When I was in middle school, that's where biology really kicked in. It was more just, dissecting things that was really influential. I went to graduate school in around 1980. Now, in the interim, over here in genetics land, essentially when I was graduating college was when DNA sequencing came about. And so I responded, really, I got an appreciation for flies. Genetics was really important, as well as the idea of doing DNA sequencing. As an evolutionary biologist, at this time, this is the beginning of sequencing to study the process of evolution. So I was reading the tea leaves, and I said all of these things were really important. In addition, computers came on the scene. This is where we started doing desktop computing, and I learned computer coding and all of that sort of thing at this point in time. But my thesis was basically, could be done in about an hour today. I sequenced 3.5 kilobases of a Drosophila gene. This was one of those moments where Tracy was talking about that moment of discovery. I discovered this gene. I did it by analyzing the DNA and doing sort of early bioinformatics decoding of the information. And I discovered that this was actually an ancient duplication of this gene. It's a tiny thing, you could do it in a minute now, but this was my thesis. After that, when I went and did my postdoc, I started doing more sequencing. The ramp over here is how much sequencing I've been associated with. And when I came here to Penn State, I embarked on a large study of the ADH region as a way of understanding the biology of Drosophila pseudoobscura in general. One of the things that I went back to was trying to figure out how much gene flow was occurring between populations. And the short story is, the higher gene flow, if this parameter is greater than 1, essentially that means that the organisms are moving all over the environment. And in fact, that's what we see, and that brought this pattern into my radar. It basically said, hey, natural selection is operating on these different chromosomes largely because these individuals are moving across the environment, and so the only thing that can counteract that is natural selection operating in the environment. So now this sort of brings us down to actually getting involved in genomics, where the second Drosophila species to be sequenced for its complete genome was Drosophila pseudoobscura. And I thought, well, let's see if we can leverage that to understand something about the biology of chromosomal rearrangements. And what we were able to do was map the breakpoints, where these breakpoints occur along the chromosome. And what we discovered was that that there were repeated regions that would allow these different regions to pair, and essentially you could break and rejoin the chromosomes there. And then I got involved in the Drosophila Genomes Project. This is a summary of really what Sturdivant was all about. Each of these lines indicates a different gene along the chromosome, and essentially it showed that Sturdivant was right, that the genes were conserved on these different chromosomal arms, but their position had been changed by rearrangements. And now my lab is working on using next generation sequencing technologies to look at 50 different chromosomes in the population, and this is just early data looking at those rearrangements, looking at nucleotide sequence variation across those chromosomes. Where we're going is looking at a detailed analysis of those chromosomes at the molecular level using this massive next generation sequencing technologies. And I would like to end by thanking all of the people that were in my lab. The history goes on for very long, and unfortunately, if I actually acknowledged everybody that has worked in my lab, it would be 3 or 4 slides. So thank you very much for the invitation to speak today.